Okay, so for this first question, the first part talks about the breakdown of what's called the sarcomere. So what I've done is made a little drawing here of the sarcomere, and I'm going to just label a few things. So this line right here is called the M line. This line here is the Z disc. What we have here, these strands down here, these are strands of actin. These strands right here, on these strands we have these projections right here that are called myosin. And these myosin are on strands of titin right here. So this is kind of the breakdown of the anatomy of the sarcomere. So this is what is in each of our muscle cells. All right, so what I've done here is I've just zoomed in right here on this actin, and I'm going to draw and label a few things that are on this actin molecule here. So this whole molecule, this whole distance is called the F-actin. Here we have a double-stranded G-actin, is what these are called. And then there are a few other things on this strand of actin that I'll go ahead and draw and label. So here in red, I am drawing myosin binding sites. And so if we look at this picture here, we have the myosin here, and on these strands of actin, we will have those myosin binding sites. And something running along these strands that kind of covers up these myosin binding sites is called tropomyosin. And on this tropomyosin, we have these little molecules, kind of look like Mickey Mouse heads here. And these are called troponin. So that is the structure of actin that we see. So if we zoom in on a little molecule of troponin here, we know these three different sites have different meanings. And so right here we have the calcium binding site. Here we have the T site, which binds to the tropomyosin. And here is the I site that binds to the G-actin chain. So as a simple description of the way muscles actually contract, go ahead and start with, here we have the myosin and the actin. So this right here is called the cocked state of the myosin. And this is where we have ADP and a phosphate here as attached, kind of keeping this in this 90-degree conformation. And so what happens is this conformation will undergo a change due to calcium and become cocked when it binds to the actin. And the phosphate that was once there will leave. This is the uncocked conformation. And when it binds to the actin here and this conformational change occurs and starts to bend, that is what will pull the actin and cause the muscle fibers to contract and shorten. So when this myosin attaches to the actin and pulls on the actin, that is ultimately what causes the muscle to shorten. So, but to get to this point, we first have to talk about how calcium plays a role. So what I've drawn here is a little bit of an outline of the neuromuscular junction, which is where we have a neuron, and this is the muscle here. So to basically explain what happens is we know that there are action potentials that get sent down neurons and neurotransmitters in these vesicles here that will be released into this junction and will bind to the receptors here on the muscle, which will then open channels that allow things like sodium to come in. And this sodium will send a sphere of influence down the muscle. And here we can draw some more channels. We know we have the voltage-gated sodium channels that can be open due to this sphere of influence and cause sodium to rush into the cell as well and propagate this sphere of influence down the cell. So what I've drawn here is what's called a T-tubule, which is basically an invagination of this muscle cell that will ultimately allow this sphere of influence to travel down and cause some changes. So we know that in the muscle cell we have what's called the sarcoplasmic reticulum here, and there are a couple of things that bind to this sarcoplasmic reticulum and also to this T-tubule that will be influenced by this calcium. So I'll go ahead and zoom. Okay, so we have the SCR here and T-tubule right here. So I went ahead and zoomed in on that just to kind of show what is going on. So here we have a voltage-gated calcium channel, and here is a mechanical calcium channel. So we know that in the SCR there is a high concentration of calcium, and this is the calcium that is going to end up causing the muscle contraction that we're talking about. So if we go back here to this process here, we see we have the sphere of influence traveling down, and it will continue to travel down the T-tubule. And so what's interesting about this voltage-gated calcium channel, the sphere of influence will travel down the T-tubule, and we'll get to this channel here, but this channel isn't actually used for calcium. This is kind of just a monitor for voltage. So this positive voltage is going to come in and open up this channel here because of that sphere of influence. So this channel is going to be opened up, as well as this mechanically gated calcium channel. And so because of that opening, this calcium is going to flow out of the sarcoplasmic reticulum, and you see how there's a little bit of a gap here. It's going to flow out of in between the T-tubule and the SCR, so down into the cell, down into the muscle. So staying here on the sarcoplasmic reticulum, I'll draw here, we have what are called circopumps that will uptake some of this calcium back into the sarcoplasmic reticulum. So you know the calcium will go down into the muscle to cause contraction, but the circopumps will take it back into the sarcoplasmic reticulum. So we know that we will see calcium come out here in between the T-tubule and the sarcoplasmic reticulum, and it will come down into the sarcomere. So for an example, we'll have the neuromuscular junction up here, and we will see the sarcomeres down here in the muscle. So that is where this calcium is headed. So I'll go ahead and talk through that. So if we go back here to actin, we see that there is a calcium binding site. So what will happen here is the calcium will come in here and it will bind to this calcium binding site. And what that will do is that will cause this troponin, this green molecule here, to uncover these myosin binding sites. So say if we have one down here, I'll just draw this here. We have our tropomyosin, and say our troponin, say our calcium comes in and binds to this troponin. That will ultimately remove the tropomyosin that is covering this myosin binding site. And so that is what we were talking about before with the myosin here. This myosin, with this myosin binding site open, this myosin will bond to this myosin binding site. And so we know when that happens, the myosin will undergo a conformational change. And this change, this going from cocked to uncocked, will pull on this actin and cause the muscle to shorten. And so just kind of an overview of how calcium causes this muscle contraction is there is a signal that goes through the neuron into the muscle, sends down a sphere of influence to the T tubules, which opens up this voltage-gated calcium channel and mechanically gated calcium channel. The calcium will leave the sarcoplasmic reticulum out into the muscle cell. The circulopumps will take some back into the sarcoplasmic reticulum, but this calcium will come down to the actin. It will bind onto the troponin, which will remove the tropomyosin, uncovering the myosin binding site. The myosin will come in, bind to the myosin binding site, and change its conformation from cocked to uncocked, which will then shorten the actin filaments, which will cause a greater effect of the entire muscle being shortened and contracted. Okay, so now to end this question, talking about the sources of energy that can cause muscle contraction. So first we will start with systolic ATP. And just a few things about this source of energy is found in the cytoplasm. So there's excess amounts of it and it's available. It is also anaerobic because it is in the cell, does not require oxygen. This source of energy contributes to muscle contractions that are short-lived, usually about 1 to 5 seconds, and not good for long contractions. So this is good for like your maximum muscle contractions for a short amount of time. So a good example of this is the muscles of the eyes. Because the eyes move very quickly, and those contractions are short-lived but are powerful. So our second source of energy we have is creatine phosphate. So this is another anaerobic source. This is what happens when this ATP, the systolic ATP, runs out, is creatine phosphate will donate this its phosphate in order to go from ADP back to ATP. So it will replenish these energy sources. And this energy source is good for about 10 seconds. So also another short-lived energy source meant for faster, stronger muscle contractions. And creatine phosphate is a supplement used by weightlifters and people that are into weight training in order to help them replenish this ATP. Third, we have glycolysis, which is yet another anaerobic source of energy. Glycolysis is the breakdown of glucose. And this breakdown of glucose provides the greatest source of ATP. For exercise and activity. So the energy source provided by glycolysis and the breakdown of glucose only lasts for about 60 seconds or a minute. Before fatigue starts to set in. So the last source of energy we have here is the aerobic or oxidative respiration, and as the name implies, it requires oxygen. And this is what happens when the mitochondria is able to provide a constant source of ATP for a long period of time. Such as going for a long run or a hike or things like that, where we are exerting ourselves for more than a minute or a short amount of time, like examples before with weightlifting and things like that. So different from glycolysis, this process chooses to use fatty acids as its fuel. And like I said before, this energy from this process can last for several hours.